All right, we're back again, and today we're going to be joined by Ka a little later on. He's at the gym, and I'm going to continue with what are we doing today? Let's see. We've got the
Cell
respiratory tract,
biology. Yes, sir.
cell
You go ahead.
biology. Yeah, go ahead.
Oh, and I pass it back to you.
Okay, enzyme activity and inhibition, and cell biology of epithelial secretion. So we've decided that we're going to be a little bit more structured and have a plan going into this. So let's see how it goes today. All right. So we'll just do a bit of solo study until we get the car back, and then we might jump into some group stuff. So until then, it'll be quiet, muted, and feel free to study along.
But I got a question though. Are we supposed to do it for like 20 to 60 minutes?
It just depends. I mean, 60 minutes we can cover all three, but if you feel like that's too long, what are you thinking?
In terms of solo study?
Mm-hmm.
Bro, I just want to get down to like this solid 150 Anki cards and I'm happy.
And how long does that take?
I'm at right now like an hour.
Right, so it'll take an hour?
Yeah.
Yeah, let's do that. So
Awesome.
1 hour mute. Basically, let's treat it like a Pomodoro, I guess.
Yes, sir.
Sounds good.
For that, I'm just gonna quickly check on I got video clips I gotta take today.
What's going on, guys?
Oh, big boss is back.
Bro, what's going on? How'd you guys go with the study today?
Going on, going good, going good. We're just doing some
Good,
solo
man.
right now.
Nice.
Yeah.
So we each pick a topic, right? Is that right?
No, just all three. Smash them all in 1 hour.
Okay,
Yeah.
okay. Let's see.
And yeah, we'll discuss, as in, like, we'll get into the group stuff. We'll go to med boot camp first, because I think those were that starts off with some easy questions.
Yeah, that sounds good.
And then from there, we will go to the med school quiz, and we'll just generate like another mid-year exam. We'll try and do that. And then
Yeah.
we'll come back and do some short answer stuff, or blue link as well, like
Yeah,
the
that
relevant
sounds
stuff.
good. I think osmosis might also even have some questions as well, if we want to do. I think they might actually.
Wait, say that again?
I think osmosis, they might have some questions in there as well.
True. Yeah.
But we'll see. All right, let's do it. So we're doing, okay, cell biology. Okay, that sounds good. All right, I'll just mute myself.
Okay.
Yo, Drew, that reminds me, we should probably send those emails, eh?
Yeah, true. We'll do that at the end of today.
Done. Bro, you remember that clavulanic acid question we did yesterday? I literally am doing the Anki card for it right now.
Nice. What's the answer? Was it just the
Okay,
beta
well,
lactam rings?
I'll ask you.
Wait, what
Alright.
question?
Remember that clavulanic acid question we did yesterday
Nah, I
or
don't
2
remember.
days ago? So,
It was yesterday.
cl-
It was how does it enhance the effect of amoxicillin?
yeah,
Oh. And then it's something to do with the beta lactase, isn't it?
yeah, it's an inhibitor of beta lactamase, which prevents the enzymatic inactivation of penicillins.
Ooh. Okay, I need to remember that, bro.
Yeah.
Hyaluronic acid. Okay.
Yo, I got 10 more cards. I'm just gonna go take a piss and come back.
Yeah, sounds good. I'm almost gone done with the enzyme activity one.
How are we going? Ready to move into the next thing, Car?
Yeah, I'm ready, bro. Are you guys ready?
Yeah, I think I've hit my solo study limit.
So how about you? You ready?
I'm always ready.
All right,
All
let's
right.
do it.
You got Med Boot Camp? Do you want to?
Yeah, I have it up. So
All right. Yeah, I'll switch.
I haven't actually. I didn't attempt the question yet, but we can do it together. I'll share the screen.
Yeah. Perfect.
Oh. Okay. Um, Let's see. Okay, I'll just share this and then. Can you guys see my screen?
Now we can.
You can see it? Okay. So this one's, I think these questions are related to the enzyme activity and inhibition.
Bro, let's do the first set of questions too.
All right, let's do it. Okay.
Fun.
All right. So within a protein, adjacent amino acids are physically connected by
Peptide bonds?
Yeah, I think so.
Bro, if I get this wrong, literally hang me and kick me out of med school.
Nah, don't, bro, don't. Juria, what do you reckon?
Well, I want to go with peptide as well.
All right, let's see. Oh, nice.
Allahu Akbar.
All right, let's read through this as well, maybe.
Yeah.
So levels of protein structure, you got primary, secondary, tertiary, and quaternary. And you got amino acid and then peptide bonds, yeah? Hydrogen bond is for the secondary. Alpha helices and beta sheets determined by hydrogen bond within the peptide backbone. Okay. And then further three-dimensional organization determined by interactions within R groups. So these are your disulfide, hydrogen bonds, hydrophobic, and ionic interactions. And then quaternary is tertiary subunits interacting together to form a functional protein. So within a protein, adjacent amino acids are connected by, via peptide bonds. A peptide bond is created by a condensation reaction between the carboxyl group of an amino acid and the amino group of another amino acid. All right. Okay, let's go next one. Which of the following is an example of a secondary protein structure?
B.
Beta pleated sheet. Yeah, I agree. Nice. So it's the same. So the two most common types of secondary protein structure motifs are alpha helices and beta pleated sheets. These derive from hydrogen bonding. What does it mean by motifs? No, maybe it's not important. Yeah, it's not important. We'll go next one. Alpha helices are stabilized by which of the following? Is it E? Hydrogen bond?
E. Yeah, it's E.
Right?
Okay.
Yeah.
Nice. Allahu Akbar.
An alpha helice forms when oxygen on a CO group interacts with a hydrogen on an NH group 4 amino acids away.
Okay.
Okay.
Yeah.
So alpha is stabilized with hydrogen. All right. What structure drives tertiary protein structure?
R groups.
Yeah,
Yep.
I think it's nice. That's right. So let's have a look here. Protein tertiary is the overall folding pattern of an amino acid sequence. The structure determined by unique R group combination that will fold in an energetically favorable pattern to create the protein structure. These patterns occur primarily through hydrophobic and hydrophilic interaction when certain R groups fold to the center or to the outside of the protein based on their affinity to the water-based cytosol. R groups involved, you can also see hydrogen bonding, ionic bonding, and disulfide bond, and disulfide bridges. All right. Which of the following is true about proteins?
A.
Yeah, I think so as well. The primary function is energy storage. Nah, it's not. There's like a lot of different proteins. So
That's the primary function of fats, no?
yeah, even. Is there a protein that does store things? There might be, right? Because there's so many. With the
For energy storage is specifically glycogen and triglyceride.
they say energy storage. Oh, okay. So glycogen and what was the other one? Sorry.
Triglyceride, which becomes fatty acids and glycerol.
Okay. Nice, bro. Let's go. So proteins have a wide variety of functions which are determined by their specific structure. Common function of proteins include structural support, transport, catalyzing reaction, and signaling. Energy storage is the main functions of fats. Yeah, that's what we said. And enzymes are generally globular proteins, while fibrous proteins are more commonly involved in structural support. Sweet. All right.
Enzymes are generally globular. Okay.
Next one. What is the function of an enzyme in a chemical reaction?
A.
Yeah, that's right. So enzymes are proteins that catalyze reactions, converting substrates to products. Enzymes help speed up the rate of these reactions so they can occur at a rate compatible with life. Enzymes are not consumed during reactions, meaning that a single enzyme can catalyze the same reaction many times during its lifetime. Sweet. How do enzymes change the activation energy in a chemical reaction?
They don't change it.
Yeah, no change.
No, it does. It does change it. It does change it. Wait, hold on. No, it doesn't change the free energy, but it changes the activation energy. So you know the curve, right? It goes down. If that makes sense.
Okay.
I swear it just impacts reaction rate. I don't know. Put it down though. I think it's no change. I might be wrong though.
Change?
No. Put D. Put B. I might be wrong.
Okay.
I haven't reviewed this.
You'll be right. I can see what you're confused of.
Ooh, let's go.
So this is the one I'm talking about here.
Mm.
So the overall energy release during the reaction doesn't change. Like this is the delta G.
Okay.
That doesn't change, but the activation energy does changes.
Okay, yeah. So that's what it means by stabilizing the transition state.
Yeah.
Okay. I haven't reviewed this yet whatsoever, so I don't know what's going on.
Nah, that's okay. I literally just went through this just now. That's why I knew. But
so why did you double, bro? You're still saying, yeah, let's pick C.
I mean, I could be wrong. Still. Okay, so activation energy is the minimum energy required to undergo a reaction. Enzymes decrease this energy to make it more favorable for the reaction to occur. Enzymes achieve this in a variety of ways, including bringing reaction components closer together, changing the conformation of the substrate, and or stabilizing the transition state.
So what they do is to stabilize the transition state, they lower the activation energy, but it doesn't lower the overall energy of the release during the reaction.
Yeah, that's right. So I guess this would be calculated from the energy of the initial reactants
Minus
and the products. Yeah. I was actually confused with that as well because I was like, wait, shouldn't it be like it changes because this whole thing is like changing? But yeah. So I guess it's the final minus initial, something like that. All right. Which of the following best characterizes the enzymatic function of a hydrolase?
D.
Yes. Yeah. Nice. Let's go. So hydrolase break up
Boom.
chemical bond using water. An example of a hydrolase is lipase, which breaks down fats into fatty acids and diacylglycerol with the help of water. Nice.
Wait, go down, go down. Oh, come on, my guy.
We got it.
Transferase. So ligase forms bond, transferase changes functional groups, and then translocase helps move across the membrane. Okay, remember that.
Okay, next one. Which of the following best describes enzyme kinetics?
B? I don't know anything about this, but I'm going with B.
Study of the rate of reactions. Sounds right. Let's have a look at the other ones as well.
Yeah, go with B. Go with B.
B?
Yeah.
Characterization of the function of proteins. That's not right. Measurement of the activation energy of enzymatic. Yeah. Okay. B is good. Nice, bro. You got this right. So.
The binding and the kit. Right, yeah.
Okay. All right, next one. Which of the following assumption is made in order to derive the Menton equation? Okay. Michaelis Menton equation.
Michaelis.
Michaelis, sorry, yeah.
I think I yet again I know nothing about this, but I'm assuming it's A.
Far exceeds the amount.
That's one of them. I think that A is one of them. Yeah, A looks right actually. No, the binding step isn't the rate limiting. It's the
Transition state, right? The catalysis step.
Is it called transition step? Yeah, the other one though.
The other
Catalysis.
Yeah.
Hmm? Product concentration. Yeah, go with A.
Yeah, because if we go based on that, like I think catalysis step is rate limiting. Enzyme substrate concentration should be constant, and then product concentration should be 0 at the beginning of the reaction.
Yeah. Right.
Yeah, that's right.
Yeah.
Let's look. Ooh, let's go.
Oh, 59 only too. Let's go.
This one's a hard one actually. Not many people got this, bro. Oh, let's see what they say anyhow.
4 assumptions.
Man, exactly what I said too. Come on, I'm actually a G, bro. No review and I still got it.
Yeah, that's insane how you got. Bro, I'm actually shocked.
Yo, let me get the placement. Let me get a site placement right now. Let me get that site placement right now.
Oh.
With the vape, huh?
I love how we walked into the Litco school and the first thing it says is no smoking or vaping.
Yeah.
All right, we got a few more. Which of the following is true regarding the? How do you say this again?
The Michaelis-Menten constant.
Michaelis.
Yeah, it is C.
No, it's not. It's not. It's not. That's a trick question, bro. That's a trick question.
Okay.
I think that's a trick question because it's the Vmax is on the y-axis, right? But Km is on the x-axis.
Km is equal to half Vmax, bro. I literally just reviewed this before we called. Or
No,
before
no.
we
No,
switched
not
over?
Carno's. Carno's, it's a trick question.
It's like this, bro. So Km is the substrate concentration at which Vmax is half. Does that make sense?
So it's
Okay.
equal to half Vmax.
No, but okay, it's like
You gotta explain it, Car. Come on.
yeah, I got you, bro. Let me get my
Elaborate explanation. I'm ready for this. Let me turn it. While he's doing this, I'm gonna go turn my Bukhor on.
the
Yeah, before you give the explanation, check what the answer is, bro.
okay.
No, let
Okay.
him explain. No, let's explain first.
No, we can. Wait, how do you guys want to do this? Should we check now?
Put your yeah. Do by democracy. Do by democracy.
Okay. What are we going? Okay, what are you picking, Julia?
Explain.
All right. So it's like this. Does that
Okay.
make, you see that? So this is Vmax.
Mm-hmm.
So half of Vmax is here, right?
Yeah.
So this point is Kmax.
KM. Yeah.
Km, right?
KM.
Sorry. So Km is actually this value here.
I like it.
It's not this one.
Okay. I'm gonna say
KM is equal to half Vmax, bro. That's the formula, bro. I'm still going with C. My answers will not change.
Julia, you pick, bro. It's either I'm going with B. You have to pick, Julia.
you're going with B.
Yeah, I'm going with B, bro. B.
Okay, so increased enzyme affinity. Yeah, I'll go with B as well then.
B. Okay.
Yeah.
Let's check.
Should have
Yo, you
stayed
made me
true.
change
Should have
my
stayed.
answer. You made me change my answer.
No
Yeah,
way! How is that true? What?
No, but I'm telling you, it's like this. Km is substrate concentration, so it's the x-axis, bro.
What?
Yeah, bro. So
Go through the explanation, bro.
okay.
He just did. He just did, bro.
Yeah, it's like this one. It's the substrate concentration when V equal half of Vmax.
Yeah.
It's inversely proportional enzyme affinity. What was the question?
Which of the following is true regarding the I keep forgetting how you say it. Microbrachialis.
Regarding KM.
KM, yeah.
So it's inversely proportional to enzyme activity. Enzyme affinity. Okay, Carl, bro. I'll give you a round of applause for that, man. Holy fuck.
Nah, bro. I honestly I thought it was the third one as well, but then I was like, nah. I knew it was a trick question, bro. So if you just think like it's on the x-axis, bro. It's the substrate concentration at half Vmax. So it's gonna be down like this.
Mm-hmm.
So it's this value here. But half Vmax is the value here, though, if that makes sense.
Yep. All right. Let's keep going.
Let's describe Vmax.
Oh, I think it's A, but I've been fumbling the bag too many times today after my dominant
Well,
display on that.
It's one fumble. Let's go with A.
Yeah.
Nah, you're doing good, bro. You're doing good.
Saturated. Ooh, yeah. Has to be, right?
Yeah.
A.
Reaction velocity when the enzyme is saturated. That looks right. Let's check, bro. Ready? Oh, nice. Let's go.
Go back. All right.
Sweet. What is true about first order enzyme kinetics? I think it's this, right?
Substrate concentration is the enzyme concentration. First order, the reaction velocity is independent.
Wait, shit. Is that zero order? No, it's first order, second order, third order. Nah, I think I'm getting mixed up.
Yeah. First order.
I don't know what this is. I'm gonna stay silent on this.
Is first order K1 or am I thinking of something else?
I don't know what's going on, bro.
Okay, bro, it's like this, right? So you know how you have substrate, right? Which is your so you got your substrate and your what's the other one? Substrate and enzyme.
Yeah.
And then you got that curve thing. So like,
shouldn't it be B then?
no, because it's like this, right? So wait, actually. Oh, wait. You might be right. Wait, is it? Fuck, now I'm confused.
Wait, I don't think substrate enzyme concentration. I don't think a substrate concentration ever dips below enzyme concentration. In
So
vivo
then-
or in vitro?
Yeah, you're right. Because there's more substrate than enzyme.
In vivo.
It's either A or C, bro. It's either A or C, I think.
I'm with you. I'm debating between A or C, but I don't know anything about this.
So it's like, I think I forgot which one is which, but this first order is when substrate is significantly higher than the enzyme. So that means you're going to have a lot of enzyme binding, no, substrate binding to the enzyme. And then shit.
Well, let's pick A. Let's pick A.
Okay, ready, bro? You. Oh,
Bro, come on, man.
Wait, what?
what?
You can't have the enzyme as the limit, like the rate limiting factor, right?
Yo, what the hell just happened?
So when the substrate concentration is a limiting factor, then it is first order kinetics. Okay, first order substrate limiting factor. Zero order enzyme limiting.
Bruh. So first order enzyme kinetics and first order velocity is dependent. Oh shit, that is hella confusing, man. Okay. Anyhow, we all good with this, or do you wanna?
Wait, check.
Yes.
What was C? You go up again. Check what the multiple choices were.
Independent.
Okay, so zero, it's independent.
It's like this, bro. Let me see.
All right, x-axis to
So, yeah, this is first order, and this is zero order. here.
So it's saturated, yeah.
So in this case here, substrate is significantly less than enzyme. But here the substrate is more than enzyme.
Yeah.
Let's see. Let's see
Yeah,
her.
that's
spread. Okay.
Or kinetics, which occurs when the velocity is dependent on the substrate concentration. This is going to occur in situations where the concentration of the substrate is relatively low. Eventually, as the substrate concentration increases, all of the enzymes will be bound to a substrate, and any additional substrate that's added at this point isn't going to have any available free enzyme to bind to. This is known as enzyme saturation, and at this point, the velocity can't increase anymore. You can see that here as this curve starts to plateau. This plateau, where the velocity is independent of substrate concentration, is known as zero-order kinetics. And that
Okay. Interesting. I need to review that. All right. Should we go next question or?
yeah.
Yeah, bro, I'm gonna be binging all these videos 30 days before the exam for sure, man. Like, holy fuck.
No, that was generally like.
Yo, I remember when we did the MCQ last, I think it was 2 days ago or yesterday. And
Yeah.
it wasn't A to D, it was like A, B, C, D, E, F, G, H, I. That's when I was like, all right, we can't even guess and get 25%.
Bruh.
We'll see.
Yo. What?
I think it's B, but I've been fumbling so hard, I'm just gonna stay quiet.
Have
It's
you
either
done
B
this?
or A.
B is good.
I've
B or A?
never, no, I haven't seen anything on the Lineweaver book. I don't know.
There's an equation for it. It's like y. It's a linear graph, right?
Okay.
It's y equals m. So linear graph has the equation y equals mx plus b, right?
Sure.
So something like this. And then b is that the y-intersection?
Yep.
So then the x-intercept is when y equals 0, right?
Yeah.
Okay, so then x, oh shit. And then I think the Lineweaver back thing is something like, fuck, it's like equal to v, it's like 1 over v max is the y, and it's equal to, what the fuck was it equal to? Oh, I can't remember for the life of me, bro. Km over V max. That actually sounds right, bro. Km over V max.
I think it's B.
Yeah, go with your.
Alright, man, I'm guessing B, bro. I can't remember the equation, bro. Oh, yo! Jab, what the hell?
Go down. I want to see the graph.
Bro.
Okay. Gotcha. So it's 1 over V, 1 over S concentration.
It was literally, if you looked at the video you showed, it was literally in the corner of the video and said high yield on it. That's all I remember.
Oh, you got it just from there, bro? Holy shit.
Yeah.
We were in that video for like 30 seconds, and he's got, bro, you're a genius. Holy shit.
I'm not a genius. I'm just a peon.
Bro, I swear. Nah, it's crazy, bro, actually. So the equation is 1 over V initial. I don't even know. Should I remember this equation, bro? Km over V max.
No, you don't need to remember this. You should be able to figure it out. It's just the reciprocal.
Yeah, bro. Oh, true. So then I just need to remember the other equation. Is that right?
Yeah. Okay.
How does reversibly competitive enzyme inhibition affect Km and Vmax?
B?
Why are you so quick with it? I'm trying to figure out what they're asking. Okay.
Yeah, what are they asking?
Reversibly.
Competitive enzyme inhibition, but it's reversible.
Okay.
So what? Okay, competitive enzyme inhibition means something else is competing with the enzyme and inhibiting
For
the
yeah.
function. So it's binding to the
Substrate.
active site.
Yeah.
And it's coming off as well. So that means Vmax would take longer to reach. Vmax, what?
So what we're looking at, Km refers to enzyme affinity, right?
Vmax unchanged, right?
Yeah, I don't think Vmax changes because
it's reversible.
Yeah. That's what I'm thinking. It's the affinity of the enzyme or binding affinity that changes. And if binding affinity decreases, then Km should increase because they're inversely proportional.
So binding affinity decreases, Km increases.
Because the inversely proportional relationship, right?
A
And
small Km means
Yeah,
high.
it's B. Let's go with B.
Yeah, it's B, bro.
you might be wrong.
Nice.
Yes.
No, you're right, bro.
Let's go.
Do we guys want to read through this or
No, Shahab got it.
Okay, let's go, bro.
Wait, go back. The uncompetitive and non-competitive and irreversibly. So uncompetitive, non-competitive decreases Vmax by reducing the amount of effective enzyme.
Active.
Km remains unchanged.
Wait, non-competitive. I get this one, irreversible competitive inhibitors. But what the hell is this? Non-competitive inhibitors. Oh, because it binds to the allosteric site?
Yeah.
But then won't they fall off? Or do they just stay there?
So if they bind to the allosteric, sorry, go, Drew. You can.
No, I think all I remember is they still alter it. So it becomes like, like then the enzyme doesn't function like on the actual site.
Yeah, so when they bind to the allosteric site, it causes a conformational change in the active site. So the enzyme itself is the thing that's not functioning. It's not necessarily the binding affinity, which overall affinity doesn't change. It's rather the fact the enzyme itself is not effective anymore.
Okay, no, that makes sense. All right, we'll go. Are we go next one or?
Yeah, go to the next one.
Irreversible competitive enzyme binds to the active site.
No,
it can't be the first one because that's competitive.
So D, right? Oh wait, C. Maybe it's not permanent.
Nah, it's because it interacts with the active site, right? So this is saying it's not
competitive. My bad. Yeah, competitive.
Yeah.
Okay.
So I think it's D then.
D. Yeah.
Because if it's a competitive enzyme inhibitor, then it should compete with the substrate for the active site, and this should irreversibly permanently. Check the answer. I don't know.
Yeah.
Maybe
D is right.
Screwing shit on my ass.
Yeah, let's go, bro. Let's check. Oh, nice, bro.
Let's go.
In irreversible competitive enzyme inhibition, the inhibitor forms a covalent bond that permanently alters the enzyme's active site. Due to this, the enzyme becomes permanently unable to bind to the substrate, effectively removing it from the reaction. Nice, bro. That's good, bro. Do we want to read these one as well? Or?
Up to you.
No, we'll skip it. Let's see next question, bro. Which of the following is true about non-competitive enzyme inhibition? Okay. Non-competitive enzymes. I mean, it binds to the allosteric. Oh, shit, you were fast, bro. I was still thinking about the question, bro. Holy shit. Okay. Wait, let me see. It cannot be overcome by increasing substrate concentration. KM.
Actually,
Wait, how did you
Because
get
the non-competitive inhibitor, KM remains unchanged, and it's Vmax that changes. We did that like 2 questions ago. And the enzyme it doesn't reversibly interact with the enzyme's active site. It binds to the allosteric site.
yeah, but that's
Yeah. Oh yeah,
Mm.
that's how. Okay, no, that makes sense. So then it must be A. So it cannot be overcome by increasing substrate concentration.
Yeah, substrate concentration doesn't affect it because the enzyme itself is changed.
Mm.
the
Okay.
enzyme. Yeah. Okay. Yeah.
Okay, let's have a look. Nice, bro. Let's go.
Man made a comeback. I was fumbling so much. I was fumbling too much. Man came back.
Bro, these are nice questions though. I hope they give us like these questions in the exam, bro. That'll be sweet.
Yeah, I have to highly doubt it.
I think the high yield one, like that high yield one, they'll pop up.
Yeah.
Since okay, sweet. Go next one, bro. Oh wait, key takeaway: increasing substrate concentration cannot overcome. Yeah, non-competitive enzyme inhibition decreases Vmax and leaves Km unchanged. Okay. Holy shit. Okay.
Let's just zoom out. Oh, no, you just have to scroll up and down.
Can you guys see that?
Yep.
What are the options?
So Vmax is unchanged. Okay,
Yeah,
then
Vmax unchanged.
It's
Um,
KM that changes. I'm thinking it's A.
So it's inhibition, reversible, competitive. Yeah. Okay.
reversible competitive inhibition. So this one Km remains unchanged, but Vmax changes. Okay. Wait, if it's if it's irreversible, then the Vmax would decrease, right?
Yep.
Yeah.
Okay, so yeah, that's sweet.
Did we get it?
Yeah, we got it, bro.
Yo,
What's the
save
explanation?
that- Save that table. Double reciprocal.
I'm screenshotting this table and sending it to the chat.
Yeah, that'll be good. I'll zoom in maybe.
I got it.
You got it? Sweet.
Yeah.
All right.
All right, last question.
What type
Inhibitor. Oh, this is probably in the table.
Yeah, I just said the table is uncompetitive. I just looked at the answer.
Oh, you did? Sweet. So uncompetitive is just 2 lines.
So that means both are affected. Both are decreased. Can you scroll down?
Yeah.
Uncompetitive, bind directly, decrease both. Yep, cool.
Sweet, bro. Okay, I think, do we want to do this one or do we want to skip this one?
Might as well keep going. Is this relevant?
Which of the
Yeah,
following-
it is.
It is?
All right, let's do it.
Which of the following best describe the function of most membrane
It's
proteins?
B.
Regulate regulated passage of molecules and signal. So it's not breakdown of waste products. Because that's like, wait, is it? Oh, it's B? Okay. Jareer, what you reckon? B, yes? Or,
I'm happy with B.
okay, let's go with B. Nice.
So lysozymes or break down toxins and waste products. Cell membrane is mainly formed of phospholipids.
Mhm.
And then the key takeaway is many membrane proteins facilitate the regulated passage of molecule, nutrients, and signals across. Also, these are like your transmembrane protein, like the sodium, potassium, ATPase, and that, right?
Yeah.
Okay.
All of them.
Yeah.
Mm-hmm.
Okay.
Yeah, because you have integral and peripheral, and this is talking about integral membrane.
Sweet. All right. Which type of membrane protein facilitates the passive transport of molecules? So passive,
Channel?
no energy. Channel.
No. Yeah.
Yeah. That looks right. Yep. So membrane channels uses passive transport to transport substance down their electrochemical gradient without using energy. What are the transporter? Transport proteins use energy, okay, to move substance against the electrochemical gradient. This would be a form of active transport. Okay. I haven't heard of anchor or anchor before. Oh, it's just okay. It helps maintain this structure and form connections with neighbor. Okay. Which of the following is true about secondary active transporters? They use electrochemical gradient, right?
Mm-hmm.
So
Wait,
it
one
can't be
sec. It's B.
Yeah.
driven by previous. Yeah. Nice. This is the explanation here. So this is electrochemical gradient, I believe. And cold transporter, yep. That's the sodium glucose cold transporter. And then this here, active transport use energy here to substance against the chromatographic gradient. A secondary active transport couples the passive movement of a substance down its with the active movement of another type of substance. This way it takes advantage of a gradient previously, blah, blah, blah. Sodium glucose co-transporter, which uses massive amount of sodium, movement of sodium into cells to drive active transport of glucose into cells. But it doesn't use ATP though, so okay. All right, are we good to move on?
Yep.
Yep.
The sodium potassium pump is an example of. Wait, don't tell me
Primary
that.
active.
Here. Bro, you're so quick, man. All right.
I might be wrong though.
Wait, let's see. Sodium, potassium. Oh, it's the potassium, 3 potassium in, 2 potassium sodium out, right?
Isn't it 2 potassium in, 3 sodium out?
Oh, is it? Okay, yeah, 3 sodium out, 2 potassium in. Okay. Primary active transport. Yeah. And it needs ATP. Yeah, I think it's primary active. Should we check? Oh.
Mm-hmm.
Okay. Cool, let's go, bro.
3 sodium, 2
3
potassium.
sodium. Yeah.
Because there's a higher sodium concentration outside the cell, and there's higher potassium concentration inside the cell.
Yeah.
Because I know that the potassium is also used to you block potassium pumps to result in depolarization of calcium to release insulin, and then it's also used in the neuron or the movement of the active potential, and then sodium is always outside the cell because then a lot of the saline solutions have high sodium concentration, like 154, which is normal saline, 154, sodium 154, chlorine.
Ooh.
Wait, 154 what?
For normal saline, the concentration of sodium is 154 mmol, I think. And then for chloride, it's 154.
Okay.
I might be wrong.
Let me check that. No, saline concentration.
D.
Yeah. Because this is a antiport, right?
That is, it's not antiport. That is primary active, because I think antiport, synport, and uniport are only secondary active transport.
Ah, so those three are secondary. Okay.
You could check though, I might be wrong, because I literally just did this in on-cue review before getting on this call.
Nah, I trust you. Okay, so that's not a antiport. All right, Jure, did you have a chance to look at the question?
The one before?
No, this
Oh,
one.
this one?
Yeah.
I don't know what a symporter is.
So symporter transport things same direction. Antiport
Okay.
is like opposite direction. And then uniport is one thing goes in. I think I explained that terribly, actually. Have
You explained it properly.
Did I?
It's exactly what you said.
Yeah.
So one thing goes in like passively, or what?
So they
No,
use the
Yeah.
secondary. They use like the gradient formed by active transport to
Mhm.
move one thing in, which is uniport, or one thing in one direction. Symport is you're moving two things together. For example, in the gut, I think you use sodium and glucose, which both move down sodium's gradient to bring
Yeah.
glucose into the cell. And then
Yeah.
antiporter is when you're moving two things in the opposite direction.
So that would be like the wait, would sodium potassium be an example of that?
Sodium potassium is just primary active transport.
Oh, okay. So okay, because I'm thinking because they're going different directions, but yeah, you're right.
Yeah.
Okay, got it. Yeah. So the glucose moves with the sodium. Nice.
Yeah. Sodium. Oh wait, it is a antiport, bro. It says.
Oh, you're right. I might have been confused, but yeah, you're right. It's an antiport. I'll remember that. That's my fault.
Nah, yeah, that's good. That's good. Because now it's gonna be stuck in my head. Nah, this is good, bro. So symporters' other name is co-transporters. Transport two types of substance in the same direction across the membrane. Yeah, that's a good definition. And then G-coupled protein receptor, that's to deal with the insulin, I remember. When bound to an extracellular ligand, it will activate downstream signaling in the cell. It is a receptor protein. All I know, bro, is that eventually you're going to get the GLUT4 transporter into the membrane. Is that right? For the G-coupled.
GLUT4 is
Yeah.
associated with insulin, so it's insulin sensitive. So when there's a pathway that results in GLUT4 being recruited onto, I think, muscle and adipose, and then GLUT2 is insulin independent, which allows the insulin to bind, or sorry, glucose to bind to result in.
Okay. So, but you need insulin to make the GLUT4, right? To recruit the GLUT4 into the membrane of the muscle
Yeah.
and adipose. Okay. And does that it's got something to do with the G protein couple receptor, right? Or
GPCR is glucagon, insulin is tyrosine kinase.
okay, that's why I mixed up. Okay. So this is glucagon. Okay. Ooh. Do we want to do these as well? Or?
What's this? Receptor.
Ligand gated ion channels.
I mean, I haven't done this stuff before, but if you guys want to, sounds good.
We can.
We can give it a shot. I haven't done this as well, actually.
Let's go.
Which
Which
of
of the
those
following?
again?
We know it's not A because transcription factors are intracellular in the nucleus.
Throw
Ligand gated.
G.
What's a ligand-gated ion channel again?
So a ligand gated ion channel is a type of channel where you have to have the ligand bind onto the channel to allow the substrate or the ions to go through.
Ah, so okay. So it needs a ligand in order for it to work.
Oh yeah.
Yeah, because there's
allow-
ligand gated, mechanical, and there's fermion voltage gated.
I see. Okay. So in that case,
It's most likely D. I might be
Hmm.
wrong though.
they act as both a receptor and a channel.
Yeah, that makes sense.
Or is it like as in like receptor for the ligand and then channel for the ion? Is that right?
Yeah.
Okay. I'll go with that as well. Yeah, that's right, bro. Nice.
So receptor.
So ligand-gated channels, also known as channel-link receptors, allow for regulated flow of ion across the membrane. These proteins have receptors that can bind to ligands in the external environment. Once activated by the ligand, the channel portion of the protein opens to allow for the flow of specific ion. Is it always like open, or is it like it can be different sometimes? Like where the ligand
It only opens when the ligand binds to it.
Ah, I see. Okay. That makes it easy to remember then. Once activated with ligand channel portion open and allow for specific ions. Okay, that's cool. And then nuclear receptors can let's just say. Is the chloride an example of this? You know, in cystic fibrosis, how there's the iron
CFTR channel.
Yeah, but that's not a ligand gated though, is it?
It's ATP gated Cl minus
ATP
channel.
gated Cl minus channel.
Or ATP. I think it's ATP activated or ATP gated. It's one of those two.
Okay,
It's
So
most likely ATP activated.
ATP activated. Yeah, that sounds right actually. Okay, are we good with this, or should we? Is there more that we should?
So wait, if it's ATP activated
ATP gated. Sorry.
or ATP gated,
Yeah.
so that's still ligand gated then, right?
Check.
Does ATP count as a ligand?
I think anything is a ligand as long as it like.
It is. He's right. It's a ligand gated. It's the only one. I can guarantee that's going to be on the exam.
Yeah,
Wait, so the cystic fibrosis one?
Yeah.
Okay.
So in this case, ACH acetylcholine is the ligand.
Yeah.
Okay. So ATP binds to the acts as a ligand, binds to the receptor, opens it, and allows chloride to flow through whatever direction it's in. Okay, now that makes sense. Oh, wait, insulin, right? Tyrosine kinase?
Yeah.
Is it? Tyrosine.
But what type?
Ooh,
I think enzyme linked.
I don't even know what these are, bro. What does this mean? What's enzyme
It's
linked, bro?
Is it just
It's a
'cause it says
receptor.
kinase?
Sorry. Yeah.
Okay.
Love it. So we're going with B, yeah?
Yeah.
Oh, it's right. It's right. Nice. So enzyme link receptors. What? Wait, what?
What happened?
I'm kind of confused. So enzyme link receptors are receptors that have a receptor portion and an enzymatic portion.
Yeah, because so what you'll have for insulin is you'll have an alpha subunits, the 2 alpha subunits on top, and then the 2 beta subunits. The 2 beta subunits are the ones that autophosphorylase the tyrosine kinase domain. So you just imagine like different subunits of the receptor. If that makes
Ooh.
sense.
That does make sense. Okay, so there's like 4 different binding sites. 2 alpha, 2 beta. Is that right? What did you say?
Yeah.
And then the beta one is the one that binds to gets phosphorylated, so changing ATP to ADP. And then the other portion is where insulin binds. Is that right?
Yeah.
So would that be the receptor portion? And then the enzymatic portion is the ATP?
Yeah, basically.
So you need the insulin binding, and then the ATP comes next. Okay. And then this.
What do you mean ATP comes next?
Like once the insulin binds, and then ATP phosphorylates the receptor.
So the tyrosine kinase, so kinases, remember, they're the ones that donate phosphoryl groups. Because phosphorylases are the ones that remove phosphoryl groups. So in this case, it's tyrosine kinase that's phosphorylating the target.
Oh shit! So it's all the way around. Okay. All right, this one's pretty good. Yeah. So are we good to move on? Next one? Or?
Yep.
Okay. Nuclear receptor.
C.
I don't know.
I don't know what a nuclear receptor is. Oh, is that like on the nucleus? A receptor on the nucleus?
Yeah, it's in the nucleus. And the only one here that's lipid soluble or is cholesterol based is estrogen.
Okay, that makes sense. Yeah, it's right.
Okay.
Okay. So the main takeaway is the steroid receptor. What? That's the main takeaway? The steroid receptor is an example of a nuclear receptor. Okay. Opioid receptors are coupled
Are
to
all
which?
steroids lipid soluble?
Yeah, I think so.
Because they're all cholesterol based?
Yeah.
Okay. So all steroids are lipid soluble because they're all cholesterol based. Okay. Opioid receptors are coupled to which G alpha subunit? What the hell?
I don't know the answer to that one.
G alpha.
What's a opioid do again?
Opioid,
Yeah.
it has to do with pain modulation. I remember that. So opioids help to decrease the sensation of pain.
Okay. So
Yeah, pain relieving medicine.
pain relieving.
Yeah, but okay.
So that means these opioid receptors are coupled to which G alpha subunit? So I stands for. I'm trying to guess what these letter means. Alpha. Oh, there's probably no. Yeah, that's probably
We'll
not going to
just
help.
pick one. We don't know what we're doing here.
Yeah.
I don't think any of us
We
have
just
done
guess
this.
one. What do we guess?
Yeah,
then?
you said-
Guess that one. I don't know. I don't want to say GQ, but GI would be fine.
GQ. Let's go GQ.
You can probably be wrong. We'll see. Yeah, it was GI.
Oh, so it's a G protein coupled receptor. Holy shit. A membrane proteins with 7 transmembrane domain when the external binding binds with an internal G protein. Also, GI is internal G protein. These proteins are heterochromatic with an alpha, beta, and a gamma domain. And active so GI alpha subunit that inhibits cyclic AMP production.
So GI inhibits CAMP, GS stimulates it, and then GQ is activates PLC.
Okay, that makes sense. All right, we're almost there, boys. We're almost there. Oh shit! What is this? Did we not do this? Enzyme kinetics.
I think it's C.
C. Maximum effect that a drug can produce. Yeah? Yeah, that looks right. Let's go with that. Yeah? Or what do you reckon, Julia?
Which of the following efficacy concentration required to produce an effect? Concentration required to produce efficacy. Okay, yeah, C.
Nice, let's go. So efficacy refers to the maximal effect of a drug. Potency refers to the drug concentration required to produce an effect, often measured by the effective concentration 50, EC50. So that would be this concentration. That's potency. Oh no, wait, of the maximum effect. Half maximum, which is the concentration required to produce 50%. Oh, EC50. Oh, okay. So EC50 refers to it's equal to the maximum, half of the maximal effective concentration. What?
Mm-hmm.
Which is the drug concentration required to produce 50% of the maximal effect. Okay, yeah. Okay, that makes sense. Okay. All right, next question. What type of drug is shown in the graph? Just the receptor activity in the presence of androgen. It's with the drug alone.
I don't get what partial is, so I'm just going with A.
I think I'm guessing A as well, but
So wait, so orange is a receptor activity in the presence of the endogenous ligand,
yeah.
Mhm.
and B is with drug alone.
Yeah.
So it's decreasing
Decreasing
Vmax.
the... No, is that Vmax? Oh yeah, it would be, right? Vmax.
Yeah, NKM. I would say inverse agonist, but I might be absolutely butchering this right now. Put full agonist because you both said that.
Let's have a look. Fuck, we're both wrong.
Partial, bro. What is partial? Bro, that's rubbish.
Response maximum percent concentration. So an agonist, let's first have a look at the agonist. Agonist causes a physiological effect when bound to the respective receptors.
So it's just
affecting the
sort of
receptors.
plateaued. We didn't have a plateau.
Wait, but
Okay, so it still has the effect, but it's not as the response isn't as high. Whereas inverse agonist is that the response is the opposite. The full agonist is you get the normal response.
Come again?
No, I think wait, you go up like Shahab,
Oh, yo,
I think you're reading
guess
the
who?
yo.
Wait, what?
My guy.
Yo, Adi! What the hell? Is that Adi?
Yeah.
Yeah,
Bro, he should join us.
yes, sir.
We should send him the link, bro. Yeah, send him the link. Get him to join us.
Addy, just message and let us know if you want the link, and we'll hop you on this right now.
Yeah, actually, that'll be sweet.
So wait, what, Drew? What did I get wrong?
Wait, so blue is the drug alone, right?
Yeah.
And then orange is with the presence of the ligand. So it's more, there's more activity with the ligand, right? It would only be in, like, inverse would be if it was the other way around. That's why I was assuming you might have read it wrong.
Yeah. No, this is a pretty straightforward question, but yeah, you're right. All right, done. It'll be next time for Addy. But wait. Go back. We're still discussing this question, call, bro.
I thought we're done already. Okay, we'll go back.
Remember that drugs mimic endogenous. If there's less activity, that means the drug is not occupying as many receptors. We're discussing partial, full, and irreversible agonists. So then this is the addition of a partial agonist, which results in a partial effect or response of the enzyme or receptor. So how I'm remembering it, right, is you have full agonist, which just results in a normal response. Partial agonist, which has a response, but it's not as much as the full agonist. It's just decreased response. And then inverse agonist results in the absolute opposite response. If that makes sense?
Right. Yeah, okay.
That makes sense. Do you have like a graph that I can look at? Or
Graph, I got you. So
Yeah.
I'll make one right now.
oh, really? Sweet.
So you have this function, a normal drug function. So full agonist right here on my camera. This is full agonist. So I believe full agonist should have just the normal response curve right here.
So that's full
put this
agonist?
later. Yeah, that's full agonist. Partial agonist is you get less of the response, but you still get a response. Like that.
And what's the black one? It's just without anything.
Yeah, that's just normal. That's just a normal interaction with the receptor. And then with irreversible, you get something like this, where it's in the opposite direction.
Inverse.
Yeah, that's why it's inverse. It was a pretty easy question, actually. Shouldn't have fumbled that.
Partial.
Yes. So we're doing the mid-year mock after this, yeah?
Yeah.
Yeah, I'm ready to get bodied.
All right. Back to it.
What the fuck is this?
Zoom out a bit so you guys can see.
There's too many words now.
Yeah, Don, how do you take care?
Ciao.
Fraction down. Ligand concentration. So blue is green line is ligand plus fixed amount of drug. So you need a higher concentration. It's fraction down. Next one, measure the effects of a drug. What's the question? In a separate experience.
Wait, Addy, what time is
I
past?
think you left.
Okay.
You left, I think.
I think it's at 8, bro. It's at 8 a.m.
What's he doing at 8? We don't even have a class.
Yeah, we got nothing. So 8 to 9. Then we got the thing. Ooh, this one's got 5 MCQ.
It's locked in.
I'm so confused right now, bro. I actually don't know which one. This is all you guys, man.
Oh, just pick one. I think we're
No,
done with.
but we can reason it. We can reason it. We can reason it. We can reason it.
Okay.
We can reason it.
So blue is so green has the drug plus the ligand,
Okay.
Yeah.
and you need more ligand concentration. What does this actually mean? Fraction bound?
I think it's the amount of enzymes bound as substrate bound to the enzyme.
Oh. Okay. So amount of substrate bound to the enzyme.
I don't think it's full agonist. I think it is competitive.
This one? Competitive antagonism?
Yeah, because it's changing Km. And if you go up, right?
Yeah.
If you go up and you read it, it says in a separate experience, no receptor response is observed when the drug is administered alone. So it can't be an agonist. So it has to be either a competitive or an irreversibly competitive.
Competitive or irreversibly competitive?
And because Km is the one changing, it should be competitive antagonism. I'm either on some crazy shit right now, or I'm completely fumbling the bag. Just check the answer.
I think you got it, bro. I'm going with you, bro. Oh, bro, let's go.
Oh, let's go! Let's go!
Dude,
Let's go!
nice, bro.
Never give up! Never give up! Never give up!
That's
So
actually
you gotta treat your exams like this, man. Not just finish them in 10 minutes.
True.
Very nice. Last question.
Oh, we should screen open.
Oh, do you want to screenshot this or?
Yeah,
Yeah.
let's screenshot that.
Nice.
Okay, how would the presence of non-competitive?
I think it's B.
Indeed.
So non-competitive, it doesn't bind to that site. So potency is EC50.
Yeah. Potency refers to the ability of the drug, right?
Yeah.
Whereas efficacy is referring to the maximal effect. So potency is the amount of drug that's required. Efficacy is a maximal response. So in the case of non-competitive, you're not changing the actual amount of substrate. It doesn't matter. It's affecting the enzyme itself. So it's a change in maximal response.
Ah, I see. Okay, yeah, that sounds right. So it's only affecting the efficacy, which is the maximal response, but it's not affecting this the
KM.
KM. Okay. Yeah, that's right.
Boom, 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 boom.
Oh, bro, you're getting all these right, bro. That's crazy.
Bro, guesstimations, bro. It's all up here. It's all up here.
Nah, it's educated guess. That's good, bro. Do we want to screenshot this, or are we good to go?
I think we're fine.
Okay. So what do you want to do now? Did you want to do the what was it?
We can do blue link, or we can do the mid-year exam.
Let's do like some blue link questions and then a mid-year.
Yeah,
Alright.
let's do that.
Do you guys want to take like a 5-minute break?
Yeah.
Yeah.
Done. Okay, we'll be back.
Because I was thinking, if you want, we could also. Because what did you study just now, Julia? You were going through the respiratory, right? Or
Yeah, I was doing a bit of that.
do you want to do like respiratory questions as well? Or?
For blue link or med school? Med boot camp.
Med boot camp. Yeah. Do you want to or?
Yeah, it would have been good. I don't know how much I remember anymore after that.
So okay, wait, but so what's like one of the like questions from it that you like? I can search up.
Let's see. I think it was like respiratory tract infections.
Yeah.
Upper and lower upper and lower tract anatomy. A bit of histology, and then what else was there? I think I did a video on rhinovirus.
Rhinovirus. Share my screen again. Just let me know if it's on the. Man. Oh, that scared me. All right.
Did you share?
Yeah. Can you see that? Oh.
Hold up. There we go.
Are these type of questions or?
Rhinovirus is, I would say, it's non-envelope. It is, B.
It is unable to pass through the gastrointestinal tract cause infection. Oh, nice, bro.
Nice.
So rhinovirus cannot pass through the GIT to cause. Why can't it pass through the GIT? Because it's not enveloped.
The stomach acid
Kills
destroyed.
it.
Acid labile.
Acid labile.
So that's all picornaviruses, I think. Oh no, wait, it's the only one. Yeah, it's the only picornavirus that is acid labile, I think.
Picornavirus. So rhinovirus is are you saying it's a type of picornavirus?
Yeah.
Okay, and then picornavirus. Wait, by saying all picorna. Oh, okay, that's all right. Wait, so does that mean that the other picornavirus can survive in the stomach acid?
Yeah, I'm pretty sure, yeah.
Okay.
Double check that. Let me see.
Yeah.
Virus acid. Yeah. It's only rhino.
Okay. So rhinovirus is the only one that is acid labile. Okay. Should we wait for Shahab to go through these together, or?
Yeah, let's wait. I'm gonna do some pull-ups.
Ooh. Oh, bro, you have the thing in the back.
Yeah,
Damn, bro, try to do 100, bro.
100.
Yeah.
I think at the moment I just do 11 end of each.
11?
At the end of each 40 to 50 minutes. So it's been a while. 3 hours. All right.
Yeah. Let's try to load the other questions. Maybe we can do how many more questions do you want to do from Med Boot Camp? It depends on you guys, bro.
So how many are there in this?
So this one on rhinovirus.
Let's just do the rhinovirus ones.
Yep.
And then go over to the respiratory tract if we have.
Yeah,
if
we
they
can.
have it.
Let's have a look. I'm gonna open a new tab, I think, because it's like all over the place, I think. So what should I search then? Respiratory.
Yeah, the respiratory infections.
Would it be under microbiome?
No, go down.
Yeah.
Oh,
this one.
Yeah.
Is it introduction to respiratory infection? Are these this is the right questions?
Yeah, this will be tough.
You'd be
Most common cause of epiglottitis. This one was pretty specific. I think it's RSV. It's either D or E, I think. I can't remember now.
Parainfluenza.
Go with D.
D? Okay. All right, let's do it. We'll wait for Shahab to get back, and then we'll run through this.
Okay.
Yeah.
All right.
I'm just gonna go use the bathroom real quick. I'll be back.
Yep.
okay, what I'm gonna say might get us canceled, but I'm gonna say it anyways, okay? They need to do an experiment, right? Where you split a group of about 20,000 people or 1,000 people into about 3 populations, subpopulation groups. One side needs to experiment with conservatism, the other side liberalism, and the other side Sharia, and allow the country to rule itself, or like the population to rule itself. And at the end of the 2-year study, you see which society has the better outcomes, just to have definitive proof of what works. That's my suggestion. That's my suggestion. What do you think? You think I got canceled for saying that?
How are you controlling variables if those are the only things you're studying? You need literally the same geography, the same.
Yeah, so you put them in like an open field, right? Open field, just complete huge open field. Then you establish like a small town on 3 sides, and then you just let them go at it.
Oh, so like you could do like 3 islands, or are we doing fields so they have interaction as well?
A field for interaction, because there's always war that takes place between societies, so we want to see what happens.
Okay. So how do you decide the relative location? Let's say you have a triangle.
Yeah. You randomize it on a field. You randomize it on a field. You go, okay, you can take this part, this part, this part. Like an open, like 1,000 by 1,000 kilometer square square, right?
Square.
And then you go like, okay, yeah, and then you randomize the location for the 3 societies, and then you just put them in place and see what happens.
Okay. No comment.
Just a suggestion. Just a suggestion.
What are you guys
Good
talking about?
studies.
What's up?
We're talking nothing.
All right.
Alright.
Yo, Kai, I just checked this. I was wrong, but I'll let Shahab answer.
Epiglottis. Staph pneumoniae. That sounds like
I would
something that's.
say A.
Is it? I thought that was gonna.
No. That's pneumonia, right? No, it's not A. It's not A. That's pneumonia. RSV, I don't think it's parainfluenza, because parainfluenza is pretty rare to happen.
What is this? What does the H stand for? Helicobacter?
I think so. No, it's not Helicobacter. It's Haemophilus. Yeah.
Haemophilus. So influenza. It's not influenza. Wait, it is influenza. Right? Or was that something else?
I think it's influenza-like, because I know we were discussing this yesterday regarding cystic fibrosis, and this is one thing that does impact in cystic fibrosis. I'm thinking it's Haemophilus influenza type B, but I might be absolutely butchering it, because I don't think it's S. pneumoniae, because that's specifically the lungs. Parainfluenza virus, it's not typical. I don't think it's RSV, so I think it's between B and C, and I'm going B.
Let's go with B. Wait, I'm thinking C. I don't know why. I'm going C though. So what are you, Drea? What did you?
No, I know what the answer is.
Oh, you know what the answer is. Okay.
But wait. So, Shahab, what did you say about parainfluenza?
Parainfluenza is pretty rare, no? It's like, isn't that a paramyxoviridae?
I don't know. But yeah, I think it's also lower respiratory.
Yeah.
That's all I know. All right.
Let's go with B. I like B.
Is that the answer? Drew, tell us what the answer is, man. No fucking way! I actually got it. Oh fuck! I actually got it. Let's go.
Bro, that is insane. How you got that right? I'm actually shocked.
What do you mean, Kai? You got it right.
Nah, I just went with Shahab. Otherwise, I would have guessed C actually, but
Mm.
so the most common
RSV.
cause of epiglottis.
Rhinosinusitis.
Epiglottitis. Okay, it's infection of Haemophilus influenzae type B. Fortunately, this infection can be almost entirely prevented with HIV vaccine. And non-typable H influenza are the most common cause of otitis. Is that the ear? Otitis?
Yeah, otitis is the ear.
And then rhinosinusitis is the nose in children. Okay. Or the bronchiolitis, sorry,
Mhm.
causes croup in children. Okay.
Oh, I forgot what croup was. Croup.
Croup is
Croup's also lower respiratory.
it's upper airway. It affects the laryngeal tracheal. That name is crazy, man. Man, some of these names, bro. Like this is the longest. Okay, it's laryngeal tracheal bronchitis.
Yeah,
What a mouthful. All right.
there we go. Respiratory stridor. Gotta be epiglottitis. D.
What's inspiratory stridor?
Some like I think inspiratory would be while breathing in. There's some sort of
Sound, right?
yeah, like wheezing, I guess.
But then why would you have drooling from epiglottitis?
Would it be rhinosinusitis?
Epiglottis inflammation.
Because it can't be rhinosinusitis because that has to do with the nose, right?
Mm-hmm.
I think you're right. I think it's D.
D. Epiglottitis.
Yeah, I went more by elimination.
Let's check. It is D. Wow.
Oh my gosh, you're here.
Maria, bro, you're cracked as well.
You know, once in a blue moon.
Once every moon.
Let's
Yeah.
go.
So inflamed epiglottis. Let me zoom out a bit so you guys can see. So airflow is obstructed by the inflamed epiglottis.
Mm-hmm.
Inflamed epiglottis obstructing airway. Where does the drooling come from? Let's see what they say. Drooling is commonly seen in epiglottitis due to painful or difficulty swallowing.
Dysphagia and adenophagia.
Dysphagia, dyspnea, and what's the sound one? Difficulty making sounds. Dysphonia.
Dysphonia. There.
Okay. Which is
all right, we need options.
Oh, bro, look at this mouth, bro.
Okay. Okay, so there's like some sort of build up.
Is it peri? Is it B?
Is above the tonsil abscess. I don't know if that would be an abscess. Can you go up?
Yeah.
Abscess.
Is that where the peritonsil is? Peritonsil. So.
It looks more like it's like a build up. What do you call it? like calcification.
Yeah, I would agree.
Okay.
Tracheitis, no.
No, I don't think it's tracheitis because that would be in the trachea, right?
Mm-hmm.
So like here.
So
I
I
think
mean,
the epiglottis. Sorry, go ahead.
yeah, like between B and D. I don't know about the signs of croup, even though we just looked it up.
So croup is upper respiratory. In the
in the mouth.
trachea.
It's literally like where the tongue is. I mean, partially, it is also the back of the, like the oropharyngeal area.
I don't know. I'm still thinking B because that's the only thing that makes sense to me.
Yeah.
Did you say C
B,
or
B, B.
B? B, B? Okay, okay.
Yeah.
All right. I'm going with B, bro. Are we good? Happy with B?
Yeah.
What the fuck?
Epiglottis?
Yo,
Is that
what
thing?
did we weren't even looking there.
Is this thing? That's the
What?
That's the epiglottis apparently.
Yeah. Bro, we're supposed to know what a normal X-ray looks like. Normal radiograph.
The thumb.
so
What the fuck is a thumb sign, bro?
Is this thing? If you look at this one, you can see like a thumb thing here.
Okay, thumb sign epiglottis.
Yo, that's probably just fillings, man. We're looking at the guy's teeth.
Yeah, what the fuck?
And that was
Oh,
real
so
rookie stuff right there.
Also, we can't see the B is impossible because we can't visualize it.
On a radiograph. Yeah. That's what I was saying.
That's tough, bro.
This one we need to lock in. So this is what?
What is the likely diagnosis?
Oh, this is a child. Croup. I'm going croup because it's a kid.
That's a good guess, actually. I'm going croup as well, bro.
Yeah, I think there's like steeple sign for croup, and it looks like there's a steeple there.
Okay. What's a steeple sign?
I don't know. That just looks like a steeple. Like I'm just thinking of like this is steepling. You guys a body language thing? This is steepling.
Oh, you're talking about like this thing?
Hand signs.
Okay. All right, let's check, bro. Let's check.
Might be a fumble right here.
No, it is. Right. Whoa, bro. Oh, you have
Mr. Steeple himself.
the thing you said was the answer.
Steeple.
Bro, steeple sign. It literally says. Bro, yo, you two. You both came to the right answer, but different reasoning.
I better be getting that pass on the midterm, bro. I better be catching that pass, bro.
Alright, the final shows the narrowing of subglottic structure, otherwise known as steeple sign. Colored in blue. This is characteristic of croup, which is a laryngeal. Laryngeal. Okay. Which pathogen is most common cause of croup? Oh, that's wait.
Oh, this has to be.
Is this?
I mean,
No.
I'm just gonna go with C.
See?
It was pretty broad.
I don't think it's C. I think it's A.
A? We did
Go
this
with Dreer's answer.
Yeah.
No, don't go with mine. Don't go with mine. Hold up. So we know. What do we know? We have H influenza. We already got that for the other one, which was what was the other one? Epiglottitis.
Yeah.
So
the epiglottitis was. Oh.
And RSV is Is that lower?
Bronchiolitis. It's
Bronchiolitis?
bronchiolitis and pneumonia, yeah.
All right, so it's between C and D.
Go D.
We going with D? Yeah, I feel C is a trick. It's always there. It's always an option.
Oh, it is D.
Let's go, Drew. My God, came clutch.
Yeah.
Drew is carrying.
This is how we're gonna reason in the exams.
Yes, sir.
Yeah, so the H type B is the epithelial, and the ISP is bronchiolitis. S
Pneumonia,
pneumonia.
otitis, sinusitis, and even meningitis, bruh.
Okay. That's just like a
This
memory.
is
Yeah.
group, right?
Group, yeah. Para-influenza virus group. All right, next one. Which pathogen?
RSV.
RSV, yeah. Yep. Respiratory
Imagine we get, yes.
Nice.
Bronchiolitis, RSV.
So bronchiolitis, you have inflammation, airway narrowing, and mucus. All right. Usually affecting children less than 2 years old. And that's cool.
Oh. No signs of phragmitis. All right, so lobar pneumonia.
Doesn't it make it difficult to breathe?
It should be dull, more breath sounds, and I'm gonna say phragmitis.
This one? More breath sound.
But why would there be more breath sounds?
Because it's, like you said, more difficult. So it's like, so you're breathing harder, right?
Fair.
Okay. All right,
What
we'll go
if
with
it's hyperresonant concussion? Can it be hyperresonant?
Hyperresonance is when there's more air inside.
Mm-hmm.
And then in pneumonia, what is the case? Like, what happens? I think there's inflammation in the lungs, right?
Yeah. All right, B, check it.
All right, let's do it. It is B.
Fluid filled.
Oh,
yep.
fluid-filled alveoli.
And that's also the tactile phragmitis, which is 99. You squeeze their sides, lats. No, tactile was back and chest, right?
Yeah.
Mm. All right, what's going
So
on?
pneumothorax hyperresonance.
Yeah.
Okay.
Oh, this has got to be molecule.
A?
Wait,
have no
biliverdin, isn't that for like the stool?
No, bilirubin is stool.
Oh, wait, isn't it urine? Isn't it urine then? Biliverdin.
Bilirubin is stool and urine.
Okay. So
urine, urobilin, and
Urobilin, sorry, you're right.
biliverdin. Yeah, let's go with A.
yeah, let's do it.
Could be C. Yeah.
Also, it is
Heme
biliverdin.
catabolism.
Biliverdin is also green. Not involved in respiratory burst. Oh, yeah, we need to know respiratory burst. The cough, reflex. And that's probably an exam question.
What is it? Myeloperoxidase.
Myeloperoxidase, yeah.
Peroxidase. Myelo.
It's the last enzyme in the generation of reactive oxygen species with hypochlorite in neutrophils. It contains a heme-containing pigment which gives sputum its yellow-green color. Oh shit.
Okay. Which cell type is responsible for generation? After lung injury. It's got to be... I'm going to go with club cells. I don't know.
I was thinking club cells myself.
Ooh.
Type 2. Yo, we should... Well, 95%, bro. What is going on?
What? That's crazy. I know it wasn't neutrophil.
Yo, pneumocytes. Wait, so... Type 2... Yo, so they're in the alveoli. Why are they stem cell precursors?
I have no idea, bro.
Which one produces surfactants?
Club cells.
Oh, okay.
One sec, I'm gonna be back. I have dumplings. That'd be
Nice.
great.
Oh, nice.
No, I think type 2 also produce surfactant.
Does it? Well, let's check.
That's why I didn't pick it. I thought it was just a surfactant thing and a macrophage thing.
Yeah, they do. It says in type 2 does produce surfactant in the lungs. Type 2 pneumocytes are cuboidal cells that synthesize and secrete the pulmonary surfactant. So maybe it's got two function then, clubs. So...
I think it's multifunction because there was also one of them takes like foreign objects back as well, like back to the bronchioles.
Which one is that? Oh,
I think it's still type 2.
Type 2 does that. Okay.
I'm not sure.
It says they're located in different areas of the respiratory tract. So type 2, they're found in alveoli, whereas club cells is in the bronchioles.
Yeah. I'm looking between type 1 and 2, trying to figure out what each one does.
So the club cell can also differentiate into other lung cell types. Interesting.
Okay, so
And they can also degrade toxin.
yeah, type 2 also. All right, yeah, whatever. That's. I think we're good.
Yeah, that one's sweet. Next question.
Aspiration, yeah. I don't know.
The hell is this?
No idea. I'm gonna pick D for no reason.
I'll
Go with D. I don't know what it is. Oral flora?
Yeah.
At least we got the least picked option. Okay.
Caused by respiratory occurs when all secretion of excellent enters the lungs.
All right. So let's do the last two questions and then hit the mid-year or the blue link. What are we doing?
Let's do some blue link and then a mid-year.
Yeah, let's do it.
Should we do mid-year first? Because I think we're going to be pretty dead after the blue link.
I mean, bro, we need to let's do the exam at our worst possible capacity, so that
No, bro,
we know that it's all up from there.
Training
That's
the
not how you do it.
Yeah, I like it.
Training the mind.
All right. I think that's that left side is looking pretty dodgy. Bottom left. All right. What do we have?
My camera died.
Oh. Bronchopulmonary dysplasia, lung abscess. Oh, wait, go down again.
Sorry.
All good. I will go with. Oh, sorry, go up again. Dysplasia, bronchopulmonary. What?
I can see this.
I think it's a pneumonia.
What?
What's your reasoning?
Because it looks like the white part is in the lobes. The lobar bronchus. Because of how it's spread out, but I might be wrong.
It's a little bit remote.
I don't know.
Yeah, I don't know either.
There's
Let's go
too
with
much.
that.
There's so much happening in this thing.
Are you talking about these ones here, Shahab, or?
Yeah.
And then in the middle part, it's not as white, like here.
Hmm.
Alright, I'm happy with that. Let's check. Lung abscess.
Yeah.
Okay.
There's
the
that
fuck?
bottom left and there's the top right. Yeah.
Oh, it's
so
this
go up.
thing.
Yeah, if you go up, because you could. I don't know. Check the original one where it's not.
Yeah, it's
highlighted.
got this thing here, bro. This
Yeah.
I saw that, but I didn't know if that was normal or not.
Okay.
Let's see what they say.
Bro, we better not be getting questions like this, man. Like, what the hell is that?
Well, of course. No, I think we'll get some of these. Radiology is crazy.
So crazy.
Learning this. All right. This is the final question. Which of the following pathogen is most likely to cause pneumonia? Patchy non-oba.
B.
This was, you know this.
It's
We're
B.
going with B. Finally, it's finally S pneumonia.
What?
Bro,
What the fuck? Yo.
Lobe consolidation.
patchy opacities and single lobe. Bronchopneumonia. That makes sense, man. A single bronchus. All right.
Yeah, it's all right. All right, let's do some blue links.
Shab, you got us?
Yeah, I got it. I got it. 1 sec. Let me just put this in the corner so I remember to take this. Okay, I'm about to share my screen.
Mm-hmm.
Have it on posterior mediastinum. Ready?
Mediastinum. Okay. All right. Posterior. Nice.
Ready?
Yeah. Oh, I'm gonna zoom in.
Esophagus.
Wait, can you?
Yeah, I gave away the answer.
Can you? What's it called? No, like so. Just make it. I think it's like full width on your monitor, but the ratio is a bit.
Oh, it's a
2:1.
presentation style, right?
Oh, okay.
How's that?
All right. Yeah, that works. Yeah.
Huh.
Hopefully.
Yeah, I can see the trachea. Is that the trachea bifurcation just below the aorta?
Yeah.
Yeah.
And then the esophagus is behind the trachea, so it makes sense.
Yeah, I know it's
Yep.
esophagus.
Okay.
Nice.
This is it's a nerve.
Right vagus, I think. I haven't done this yet.
I'm gonna go with azygos. I don't know.
Azygos is a vein.
Wait, what's the? Okay, so we have vagus nerve on
That's
this
the
side.
vagus.
So phrenic
Phrenic would be going like this.
would it just be going straight up?
Yeah, it'll be hanging there because it goes into the lungs.
All right.
I'm just gonna say vagus. I don't know. What do you guys think?
Yeah, I'm guessing vagus as well.
Greater splanchnic.
So that goes to the lower abdominal area.
Bit weird. Can I move to the next one?
So where would the lesser be?
I have no clue. I have not reviewed this yet.
Oh, okay. Yeah.
That is the vagus.
That's the
That
vagus.
is
Yeah.
left vagus. Yeah.
100.
Left
Yeah.
vagus.
Is that the phrenic then?
I think
Wait,
that's still vagus.
structure. It's got to be something.
No.
I'm gonna call
Sympathetic
it.
trunk.
Yeah. It is dorsal.
Boom.
Yeah.
Thoracic sympathetic trunk.
What is this? Oh, so that's the descending aorta, and then we're going to.
Is that sympathetic artery?
No, it's a posterior intercostal.
Okay. Is there a specific one?
I think posterior. I might be wrong.
T7, T8, T10.
Oh, there you go. It is.
Oh, nice. Oh, what is this? This is going to the brachial plexus. No. I don't know what this is. But it's right next to the heart. No. Where is this? It's up here. I don't know.
Is it one of the
I
brachial
have no-
plexus or?
No, I don't think so.
All right, because that's for the arms and the pectoral.
No, we would not have fucking got that whatsoever. Cervical,
Oh.
thoracic, ganglion.
Stay like.
When did we do posterior mediastinum?
I don't think so.
Okay.
You want to skip this and do a different one then?
Yeah,
Yo,
do
what's
like
our track tomorrow?
Esophagus vein. Yeah, I got that. Our prac tomorrow is respiratory
pathogens.
pathogens. Yeah.
Oh, okay. So it's not anatomy.
Yeah. So let's do something that we would have just been doing, like resp related.
Resp related. You want to do lungs and ventilation pathways?
Sure.
Let's do it.
And we can do larynx, pharynx as well. Pleura. Oh, specific part.
I think that is. It's not parietal. I think that's visceral. Parietal. Visceral, I'd say.
So right.
No, that's a diaphragmatic pleura. There you go.
Yeah. Diaphragmatic. That's good.
Yeah, there you go. From a fumble. I caught the fumble.
Let's go.
Cervical pleura.
Yeah.
Costal pleura.
Costal's good. Body of.
I did this card today.
So there's the parathyroid.
Middle Kirkle thyroid membrane. I think it is.
I don't know then.
Yeah, there you go. I got it actually. I just did
Nice.
this card this morning.
Nice.
Oh, that's impressive, actually.
Deep to this muscle. All right, Shahab, you got this. It's the same set of cards.
Cricoid cartilage. I think it is. I haven't done this one yet.
No.
Oh, I
come
did.
on.
Apparently,
Yeah.
I did.
Cricoid.
This one's easy.
I don't know what I'm looking at.
That's a trachea.
Oh, is that the wait? That's the
So this is just a bifurcation.
bifurcation. Yeah, is it?
It's
Oh
the
yeah, because
carina.
it's
The
right next to
carina. Yeah,
the
same
aorta.
name. Yeah, same thing. All right, I'm happy with that.
Yeah, carina trachea.
Yeah,
Wait, what does carina mean?
I think it's just another name, isn't it, for
Oh,
the
bifurcation.
bifurcation?
It's a keel-shaped
The-
anatomical structure.
Uh, right bronchus.
Right primary bronchus. Yep.
Left. Ooh, can't say primary, or can we?
Secondary
Secondary
What's that?
bronchus. Still
Bronchial?
bronchus.
bronchus.
No, I don't think it's bronchial yet.
Left. Wait. Um,
Yeah, left.
left. Okay.
Left.
Wait, is this posterior?
Yeah, left middle lobar bronchus. I think it is.
Middle. Yeah, okay. No. Wait, there is no left middle. Is there a middle?
Left inferior.
one.
I think. Yeah, you're
Yeah,
right.
inferior, yeah.
Left inferior. Bronch
Bro, what is that?
completely fumbled.
Wait, because the other three are like the one like when it branches off. What's that part? Come
This. I think these are the
on. No,
inferior middle.
the first branch off, yeah, because there shouldn't be three. Bronchus intermedius. Does it have another name? Can you see if it has another name?
Intermediate stem line or intermediate bronchus.
No. Okay.
No, this is the right middle lobar bronchus. If I'm not right on this, I'll genuinely kill myself.
You're good.
Wait, maybe it's the same name for a different thing. Let's see.
I'm not gonna kill myself, but what the fuck is that? I'm not gonna say anything now. I don't know what the hell's going on, bro.
Left lower lobar, left inferior lobar bronchial.
What the fuck is that, bro?
Is that the right? See, okay, so it was posterior view. Wait, our things are in the way. Can you switch the? Because usually it says if it's a posterior view.
It doesn't say anything under it. I'm looking
Ah.
at it on my screen, literally nothing there.
I guess it should because it's we should be able to tell from the angle.
Yep. Bullshit. Oh, cardiac notch, I think it is.
Yeah, cardiac notch of the left lower.
Oh, boom. I actually got that. Let's go. Oh, shit. Or they're just giving us the answer, the lingula.
All right, so that's some fissures.
Oblique fissure.
Yep.
Yeah.
What's
Phrenic.
this? What is
That's
this?
a diaphragm. Diaphragm.
Okay. All right. Cool. Phrenic. Hemidiaphragm. You middle lobe. Hilum.
There we go. Those 80 Anki cards you did today come in clutch, eh, Drew?
That's it, man. I don't know if they were these ones, but
Let's do stomach, liver, and spleen.
wait. There was nose and throat. Is there anything there? Yeah, larynx, pharynx, sympathetic trunk. Okay.
Bro, I have not touched this whatsoever, so I don't know what the fuck is going on.
This is this week, so we might as well get a head start.
Yeah.
What is
That
this
one's
thing?
easiest.
That looks
That's
like
the
a
hyoid bone. Yeah. Greater horn of the hyoid bone.
Okay.
It's not even that I looked at the answer by accident, it's I already knew this. This is one of the only ones I know.
Wait, so is there
Oh,
okay,
come on.
so with the would there be on the other side, would that also be the greater horn as well on the other side?
Yeah, greater horn. This is the lesser horn right now, right here.
Yep.
This is the body.
Body of okay.
This one's easy too. This is styloid process. I would have got it without looking at it.
Starlight.
Yeah, this one's. Come on. This is it's 2 syllables. It's a short one. Over. Oh, I don't know.
It's not occipital condyles are right here. I don't know what that is. Pharyngeal tubercle. That's the laryngeal prominence.
Yeah, laryngeal prominence.
Kirkle.
Wait. Where's the prominence? Is the prominence the one above it?
Let's check.
I think it's the one above it.
It's this, yeah.
Yeah. Just above.
It's this right.
Yeah.
Cricothyroid. Take a look.
What are we looking at?
Bro, what the fuck is that? Subglottis? Epiglottis? I'm going epiglottis.
I don't know.
Airy epiglottic folds.
You got it.
I did not get it. Infraglottic cavity? Bro, I don't know what the fuck that is, bro.
That's the first time I've seen that.
Okay, yeah, we should get this right. This is the Vegas.
Yeah, Vegas.
Bro,
What the hell?
Superior cervical ganglion. Okay.
from the question, we should know not to call it a nerve.
Yeah, it's nervous structure, not nerve, right?
Yeah. That's usually a plexus or a ganglion or something else.
That's the last question.
Okay, that was I think
Lie.
we got probably 1 out of 7 there.
Like, let's do something we should know.
Okay,
Yeah.
you should know.
Me? We should all know this. Let's do forearm, wrist, and hand.
I shouldn't know anything. All right, yeah, maybe. Okay, I should know this. Well, I still haven't done the nerves. Okay, what nerve innervates the muscle is...
What is the back of the head?
No,
No,
it's
this
a
is the
flexor.
front. So
This side,
anterior.
right? It's either median or ulnar. I'm going ulnar. Median nerve, okay. Flexor digitorum superficialis. Median nerve.
So the muscle is the flexor digitorum superficialis muscle. Okay.
Brachioradialis, I think. Oh, boom. I actually got it.
What's this? The artery.
Is that the
Ulnar?
It's
I
yeah.
was gonna say Ray.
No, not radio.
Oh,
Ulnar.
Yeah,
oh,
we
it
got
is
it.
all. Okay.
There's your radio.
Yeah, you got it.
But yeah. No.
Median.
Now wait, hold on. Because this is posterior. This is the dorsal.
Oh, you're right. It is posterior. This would be radial.
Extends radial nerve. Yeah.
Median. I was right.
So median is on the back of the hand, like here. Is that right?
I don't think this is the back of the hand.
Oh, this is the. Isn't it?
Yo.
So it's like this. Here.
It's like this.
That thumb is confusing me, bro.
Thumb's been
Bent. Yeah. Medium.
should be able to tell from the actual vessels.
Extensor. No.
Okay, yeah, this is the
Digitorum.
back. All right, can you go back to the previous one? All right, so front of the so because we can see if you look at the index finger, you see
We can't see
the
nail.
flexors. The flexors are like they look like flexors. Okay, fair enough.
This one.
Identify the muscle. Extensor. I'm gonna go. It's got to be longest, yeah.
Oh, it's longest.
I don't know.
I think it's just the extensor digitorum. Yeah.
No. This is where the. All right,
It's
we're
like
back.
a connective tissue or something.
I think it's a palmar fascia.
Yeah.
Palmar fascia. Yeah.
Palmar
Oh,
aponeurosis.
Oh.
Same thing.
Oh, it's the same thing. Will we get full marks if we say fascia?
Yeah.
All right.
I don't think we did this yet. We don't need this one. We also don't need this one, but I think this is the palmar, superficial palmar. Yeah. We don't. Oh, we do
All
need
right.
this one.
Surely we got to get this, man.
Oh, bro.
What's scaphoid?
What's a mnemonic?
Yeah. Wait. Hold up. So that's the pinky over there. Is that scaphoid? I guess.
Yeah.
Yeah. Some lovers try positions that they can't handle. Radial.
Yeah.
Auxiliary. No shit.
No.
Yeah. It's brachial.
Radial. Yeah.
Oh, radio. Yeah. So this is the
Ulnar.
front of the hand.
Yeah. Ulnar.
Oh,
Yeah.
it's front. We're back at the front. Yeah, I'm still going ulnar. Oh, it was ulnar.
Yeah, it was ulnar.
Cool. What's this? Palmar sheet aponeurosis again.
Dorsal.
Transverse carpal. Let's not worry about this one.
Oh.
I don't think we need. I think this is thenar, but I don't think we need to worry about it yet. I don't think we need to worry about this one either.
Oh, we should get that though. Radiocarpal.
Yeah. Yeah, radiocarpal makes sense.
All right, let's do the MCQ, man.
MCQ
Before-
done.
Yeah, let's just do the exam, mid-year exam.
Oh, I'm gonna get cooked.
We're all gonna get cooked, bro.
Nah, bro. I think.
Okay, how do you want us to do this one? Do you want me to just generate one?
No, we'll just do the what's it called? From the website, we'll just do It'll be different questions for everyone, I guess. But at least
The
they'll
only
be reflected.
problem, yeah, is I don't think it'll be any MCQ, not MCQ, SAQ.
Short answer. Yeah, there won't be any short answer. So we can do that, and then if we are still willing and able, we'll do GPT short answer.
Done.
All right.
You look so exhausted, Drea.
Yeah. I just need some water.
Yeah, sure.
Yeah, I'm gonna be back too because I'm gonna go throw this out and wash those dishes.
Are you gonna? How long is that gonna be?
2 minutes. I wash dishes quick.
Okay. I'm gonna open up some. No, what is this? Why is it not showing? Mm-hmm.
Okay, I'm ready, bro. I'm not trying to go to this 12 o'clock prac, bro. I'm gonna show up to the 4 p.m. one.
Oh, you're changing prac, bro?
Who do I change with? Drew, what time's your?
I don't know. I'm guessing it's gonna. If yours is 12, then mine will be the one after.
2, right?
Yeah. Mhm.
Whatever. I'll just tank it and show up to this prac, man. F it.
We should be all right, man.
Bro, I didn't prepare for the prac.
Me neither. I'm probably still gonna go through like 130 more cue cards,
Mm-hmm.
like new cards, then 250 review, then go to bed. Gym and video. Gotta finish that video, bro. I've been lacking on it.
Mm.
Are you almost done, bro, with the video?
I'm almost done recording the clips. I just need to do 2 or more days of recording, and I'll probably be Wednesday and today. Then I'm gonna have to edit it, and the editing portion is gonna take a while because I'm actually gonna put in a bit of effort into editing this one.
What program do you use to edit?
Premiere Pro, After Effects, and Resolve. Like I do color grading in DaVinci.
Ooh. Oh, I used to use DaVinci actually.
Yo, this Twitch viewer, full respect.
But who is this guy, bro? He needs to expose himself. Like,
This
man,
person
if you leave a comment, I'll pay ID you 20, just to know who you are. Oh, I need to pray Isha too.
Yeah.
Oh, then we have football tomorrow at 8. Wait, I wanted to check one thing. What team are we playing? Like, I want to see the score. Like, what was our last game like?
Oh, are they public? All right,
Sorry?
I'm gonna set up are the scores public somewhere?
Yeah. If you go to the website the guy sent, they won 11-7 against FC Cuties.
Who's that? What's our
I don't know.
other team's name? Oh no, I've just doxed
Multiple
myself.
sclerosis.
Oh yeah.
Doxed yourself?
Yeah, I had my email and again. Actually, it didn't show. All right. Prepare for exam. It has a reattempt incorrect questions. That's pretty good. Are you gonna do it? I'm gonna start mine so I don't finish at tomorrow.
Yeah, let's go.
Okay.
Fuck, 45 questions right now. My brain is fried, bro.
I'm back. I'm awake again.
Wait, are we doing individual or what?
Yeah, individual.
Yeah.
I'm just gonna have it up so viewers can see it. Our Twitch guy.
True hero, whoever this guy is, much love to you. Man doesn't want to expose himself, but Yo, what's going on your screen right now, Jareer?
What's up?
What are you watching, bro?
Yeah, bro.
The audience is watching some med school students take a medical quiz.
Are you watching with them, or do you have like a?
No, I'm doing the quiz. I'm a med school student doing a medical quiz.
Oh, true.
I'm done.
I'm up to question 13.
13?
Yeah.
Bro, it's a 45-minute test. It's been 8 minutes. What are you talking about?
It's been 14 for me.
Oh, actually, yeah, 14. Or you can set up the short answer. We'll do like 10 questions or something.
I was gonna change a couple of my answers, and then
Okay. This one's harder than last time.
yeah, I don't know what's going on. I'm just gonna submit it and call it a day. See what I get.
I still got 20 something left.
As long as I get like a 50 above, I'm happy.
Did you get it?
Full man, 48.9. Fuck.
I had to skip like half the first 8 questions. I was like, I'm not publicly showing this test now.
Yeah, holy fuck. Okay, that makes sense. I fumbled that. Why did I change the answer? Fuck. Oh, I didn't read that properly.
I might stay at yours tomorrow
Done.
after the game.
No, I could have easily got a 60 on this. There's like 2 or 3 questions I completely fumbled just because I wasn't reading it properly.
Should have treated it like med boot camp.
Yeah.
Secondary lymphoid tissue. Secondary lymph. Thymus. I got 23 minutes.
Yo, I'm just looking at this thing they're doing, bro. I would not get any of those questions right, man. Okay, I'm gonna go do my dishes. I'll be back.
All right. Carl, how long you got left? Or how many questions?
I'm almost done. I have like 10 left.
All right. Yeah, I've got 7 left. I'll let you know when I finish.
Yeah, too easy. So how many more minutes for that?
15.
15, okay.
Bro, I feel this guy up here. I'm a first year medical student. I feel stupid watching this video. Bro, I feel like a retard watching this video, bro.
Yo, I got the 55.
My God, 55.
We made it. 0% on endocrinology.
Yeah, you're right.
Bro, I mega fumbled today, bro. 48.9? Holy.
I got one question left, bro.
Yeah, you got it.
I'm guessing this. Okay.
Ooh.
Let's see what I
Watch
get.
class stop on a 70. I'll actually start bow down to you next.
The
Wait, what'd you get, Drew?
50. Why, bro? What'd you get? 56?
55? Wait, let me see. Mine's
I
still
got
processing.
55.6.
Oh, that's a pass, bro. Let's go.
Yeah. That means you
That's
beat me,
great.
huh?
Nah,
You seem pretty confident.
Nah, bro, I'm not. I had to guess quite a bit.
Bro, of course.
Oh, I had 10 minutes left, actually. Oh, I got
Yeah.
53. I passed.
All right.
Oh, my God. Seems like the
Bro.
tables have turned from last time after I got. Bro.
Nah, bro, you did it so quick. I swear, man, you didn't use your time properly, man. You had so much time left.
I was so lazy. There's so many questions I could have got if I actually tried.
Yeah,
Mm.
like how fast did you do it in? Like 8 minutes?
12?
14.
12?
Yeah.
14, yeah.
That's
14.
insane.
So I got 100 in the interactive imaging. You know where it's like select where the chamber is or whatever. I got a bunch of those questions, so that helped.
Bro, like I caught you. Remember when we were talking about the right and left ventricle on what he's talking about, the interactive?
Right in the ventricle.
Bro, I got it wrong this time. I don't know why I got it wrong.
No way.
This time I got it right. This time I got it wrong, bro. I don't know what's going on.
Oh, is that the ultrasound, right?
Yeah, the ultrasound. It's because the right ventricle is always smaller than the left ventricle. So the left
Yeah.
ventricle has to pump out more blood. Bro, I like a
Mhm.
dumbass. I selected the bigger one for the right ventricle.
Oh. But that's easy fix though, so it's okay.
Yeah.
Man, I swear some of these ultrasound is just ridiculous. I have no idea how to differentiate.
Yeah, me neither. I don't know what the hell I was doing half the time. I don't even think we'll ever get questions like that.
I hope not, bro, because man. Actually I got some of them right, but bro, like what the hell? What was this one? Oh,
Man, I got
no way. I
20%
changed this
on anatomy.
question.
Oh, you changed one?
Oh, yeah. It was the stratum. It was like the skin layer, bro. It's so weird, bro.
Wait.
It looks so weird.
Oh, did you go with the... Wait, did we get similar questions? That was the... I mean, in my one, it was lucidum, I think.
Yeah, lucidum. Bro, what? It's actually... So A is... corneum?
Dude, this is going to be in the actual video. I picked the radius instead of the ulna. It's over.
No
for me.
way.
It's over.
My God. Oh, man.
Oh, what? It's pseudo-stratified columnar cells in the trachea?
Yeah, and I got the same things. Right? The larynx, anything in the larynx, pharynx, and trachea is pseudostratified columnar epithelium with cilia, because that's the respiratory epithelium. I got that one wrong. I brain farted that one.
Dude, I thought it was simple cuboidal. What the hell's wrong with me? I knew it wasn't columnar, because that's to do with absorption. And it wasn't squamous either. I was thinking it was F, bro, like simple squamous ciliated. I didn't see this. Oh, I didn't realize it was pseudostratified. It's kind of easy, bro, because it says ciliated at the end. I should have knew.
Yeah,
Oh, that's dumb. Okay.
that was a
So I
question
feel
I fumbled.
Yeah. And then these, bro, they asked me like, there was 2 questions on Clostridium bacteria.
Botulinum?
Difficile.
Difficile? Bro, I don't know anything about that stuff right now, bro. I'll be honest with you.
Oh, bro. Bro, I knew one. Oh, wait, they're both positive? Shit. Okay. So I got botulinum and difficile. Okay. Because I remember doing it yesterday. Okay, so they're positive. They're both positive. Oh, bro, they're both positive. They're the same. Both positive rod anaerobic. So if it starts with Clostridium, then it's positive rod anaerobic. Okay. Whoa. I said they were both negative rod aerobic for some reason. But that's good to know. I'll know for next time.
That might be the most legendary fumble.
Bro, if you spent more time, I feel like you could have got like credit or something for sure.
What is this, bro? What is this?
Pen of cells.
Hand
making
cells?
some-
I don't know.
Bro.
They are cells that produce large amount of antimicrobial peptides, such as your alpha defensins and lysozymes, and they're capable of phagocytosis.
Damn, how'd you know that?
Oh, that's just in the question. Pen of cells. All right.
Oh, that run though. 1, 2, 3, 4, 5. That's good. Oh, I gotta hide that. Oh, it was
Oh,
exon.
no, show us. In which segment of nuclear sequence is kept during RNA splicing? Exon. Bro, you gotta think like this. It's either
Yeah.
exon or intron, right?
Yeah, okay.
Right. So it has to be one of those. So you remember like this. Intron is the stuff that's in between. So they don't code for anything. They're just in between the exons. So they get cut
Okay.
out. Yeah, that's it.
All right, done.
Yeah. So they're the non-coding region. That's how I remember it.
G1 growth. What am I doing?
Oh, man, there's so many mitosis question. I need to
Yeah.
study for that.
That's good though.
Oh, this one. Wait, what
Oh,
is it?
let's go.
Is it oocyte? What is it pointing to? Pin A. Oh yeah, okay.
Right. It was round, so I picked it, man. That's all it is. Yo, the odds were crazy.
Bro, you got that. That's insane.
Oh man, this was close. Because I really thought it was ending there. Because now it just looks like
Is it the duct? No. Oh, it's a respiratory bronchiole.
I switched it from the
Oh,
respiratory.
C is the duct, I think. C is the alveolar duct. E is alveolar sacs, I think. Okay.
We haven't done this.
Oh wait, what's that one? What's
What
that
were
one?
you saying? Oh, it was squamous. Ah, man.
What's 24?
Lamina. Appropriate.
Pin D. Yeah. Okay, that makes sense. I actually had the same one, but it's asking me to identify A instead of
What is this? Oh, it's another switch. All right, good. Spleen. Yo, all of these I changed. Like these last 3 that I got wrong. This was a guess. Look at that. Beautiful.
ventricular cell. Nice.
Perfect.
Nice, bro.
Perfect.
Wait, what? How did you figure that one out?
Perfect.
Bro, go up.
Which one?
To 30, 33.
This one? Yeah, this one was.
Oh, actually, no. This one. Oh, because that's the left ventricle. Wait, how'd you figure this out, actually?
Yeah, because this one was the biggest.
Ah. And then you just. Okay. So then that must be the left ventricle, then, right?
That's pretty much it. Yeah.
And then, okay. Okay, no, that's cool. How about the next one? Indicate the location. Oh, yeah, that one's easy too.
Yeah.
Left ventricle, left atrium. Left atrium.
I think I went because I thought the right atrium would be. I can't remember how I reasoned this one. But yeah. Oh, I did get this wrong. It was big.
Oh.
Yo, come one, bro.
Wait, D is
Oh, okay, yeah. That's
come.
base.
Let's get sun. What? Come, because B is lucidium, bro. Because I have that question. It's
Lucidum.
yeah,
No, B can't be lucidum.
No, it is. It is lucidum. I have it here.
Oh.
It's stratum lucidum, which is wait, that doesn't make sense. So it's like
No, are you sure?
yeah, bro. I'll show you. I'll screenshot.
Oh, because C is spinosum.
Come, let's. Wait, is it
Or sorry. No, I don't know, man.
D? Wait, Shahab, do you know this one, bro?
Isn't lucidum only in thick skin?
So there's no lucidum in this one. I'll
B
take
is
that.
lucidum, bro. B is lucidum. I'll show you, bro. How do I? I'll post it on the Discord, bro. I'll show you guys.
Because this right here, underneath B is granulosum. That's spinosum. C is spinosum. D is basale. Corneum is A.
And then so E is dermis.
Dermis. Yeah.
Wait, doesn't it
Dermis.
go like come? Let's get sunburn. So stratum corneum, stratum lucidum. C is
Granulosum.
come. Let's get granulosum. Come.
Spinosum basale. Come, let's get some bitches.
Come, let's get some. So then shouldn't D be S? Spinosum?
D should be basale because it's right by the basal, the basement membrane, the basal lamina.
Oh, is that the okay? Is that E? Basal lamina?
E is dermis, I think.
Yeah.
Oh, E is the dermis.
Dermis, basal, spinosum, and then I don't know the other two.
Lucidum and
Corneum is A. I think B is either I don't think it's granulosum. I think it's lucidum. He's right.
Yeah,
it's
it's-
lucidum most likely.
lucidum.
So no
Oh,
granulosum.
they missed. Aren't they skipping one of the thing because they're supposed to be 5, right? Like, come, let's get some bitches.
Yeah.
They're missing.
They are.
Sounds good. Oh yes, we just did this.
Maybe
How
we'll
was
just
this
call-
intermediate filaments? Okay.
Which cytoskeleton elements resisting stress? Intermediate filaments.
I think I got an easier one this time, to be honest.
Oh, is this the philo? Wait, what is this from? A.
It is epidermis. I had this question too.
I thought it was asking E, and I couldn't figure out whether it was hair follicle or A.
E would be hair follicle, I think.
Well,
Is
even
it?
this?
Yeah.
Okay. Sounds good. Clutched it right.
Ooh, what's this? What's this? C.
This one was easy.
Oh, okay. This one's good.
Yeah.
Yeah, that actually makes sense.
I have no idea what's going on here.
What in the world?
All right, we have an explanation that does not help. 12%.
12%. Oh, that's funny.
Oh, what was this? Uric acid. Couldn't figure it out.
Oh, is that uric acid? Oh, wait.
And-
Yeah, I remember, Carl, you had this question.
Yeah, I got it wrong again. I think I got. Pyrimidines. Oh, mine was asking for pyrimidines. Sorry.
Oh, okay.
This was
Oh, that
last
one.
time. Remember last time we did this?
Yeah, I do remember that question, but I forgot the answer. Okay, it's uric acid. Okay. All right, I'll commit to memory this time. So pyrimidine is carbon dioxide.
Let's go.
And
Pretty solid. I'll take that.
Yes, sir.
Still got 8 weeks. to actually learn the stuff.
And that's exactly what we're gonna do here.
Bro, I feel like if
Yes.
we do like mitosis and meiosis, we're gonna get like a few more marks because it's like every time they have like 1 or 2 meiosis. I feel like.
Yeah, like cell
The phase.
biology and genetics is pretty heavy.
And then there's like the glycolysis as well.
Yeah.
Bro, I got asked like a few of those in my one.
Mm-hmm.
I gotta go through those again. Yo,
Alright.
I'm gonna share my screen. Can you guys see if you
Yeah.
can see
Sure.
what this one is? Let me see if I can. Oh, bro, I have so many tabs open. Where is it? Oh, it's this one, right? Can I see that?
You gotta share it.
Oh, I got that
Present.
right.
Wait, I'll present it. Okay, you got it. Yo, what's the question? Oh, nice. Oh, did I don't know if I got that one right. I had that same question.
This one, I just literally guessed it. I had no idea.
What's that?
M
M cell. M is mast cells, right?
I don't know what
Yeah.
M is.
Is K Kupffer or something?
Yeah.
Oh, this one's pretty easy. The hypochondrium.
Oh, nice. Except the
Oh,
gallbladder.
it's secretin. Whoops. Okay.
Thus inhibiting gastrin. Right, secretin. Oh, that's tough.
Man, this one I was like, yeah, no, my brain wasn't braining there. Oh, it was this one. Oh man, I changed it last minute.
Apoptosis. Nice.
P53 gene.
Sorry, did we get like it's like half of it was the same. Oh, Hardy Weinberg, come on, man. This is gonna be in every exam.
No way.
Did you guys learn this thing already? Hardy
No,
Weinberg?
we haven't done it yet. I think it was in one of today's lectures, probably.
Oh, bro.
I had this right because I remember you doing this question.
Oh, bro. I remember as well, but I can't remember which one. I knew it was one of the 50, bro.
This is in the concise notes.
Wait, it wasn't asking for pin A? I swear it was asking for pin A.
No, the answer you picked was A.
Nah, bro, I swear. Oh, they were asking for pin B, bro. What? But would pin A be bile duct though?
I don't know.
Oh, I swear, bro. Oh, bro, I swear they were asking for pin A. Oh, I don't know. I'm starting to see things, bro.
D. Oh man, you got the tough ones.
No, this one's easy, bro. This one's easy.
I've never heard of a cumulus cell, man.
I haven't either, but I knew it was that one because I did this question last time.
That's what makes it easy, huh?
Yeah, but this is theca cell. This is theca cell. I know it wasn't zona pellucida because it's like on the outside of the egg. And then this is the oocyte. B is B. This is antrum, I think, and that's zona pellucida. Yeah. Okay.
Okay. Need to do embryology.
Yeah, this one I was deciding between lamina propria or smooth muscle, but then I was like, because of the striation. No, like the
Come on,
way
we
it
should know this. Yeah.
Yeah.
That's like I think we've done quite a few of those in the labs.
Oh, which one? This one?
Yeah, the histology labs.
Oh yeah,
Like,
yeah.
lamina propria a bunch of times.
Yeah.
Muscle as well, yeah.
This one I guess, bro. I didn't know this. Oh, it's a Kupffer cell. Okay. This one, oh man, this one was,
Bro, what the fuck is that?
bro, I had to. Okay, let's try work this out, guys. Okay, is 3 like the heavy chain? It's like it's an immunoglobulin. Or maybe 2 is the heavy chain, and then 1 is the light chain.
Bro, I don't think so. Scroll down.
Yeah.
What were the answers?
It's option 4.
Oh, M cells are not mast cells, by the way.
So
M
What is
cells
it?
are microfold cells.
Microfold cells.
They're in the Peyer's patches.
Bro. Oh, Pire patches. Oh, so that's in the. That would be like. Pire patches is in like the GIT somewhere, isn't it? No, wait, actually.
Yeah.
That's on the. Wait, yeah, it is, right?
Oh,
I can't remember which one it's in.
I think it was small intestine. I don't know.
I'll have to review that later. Anyways, so this is heavy chain. I got that right. I got that right as well. So I knew these 2, but I didn't have enough time, so I just guessed it. But I knew this was heavy chain. I knew this was light chain. Or it was one of these 2, but FC region. This is the FC region. And this is the Fab region. Where was the option? Option 4 was right. Heavy chain, light chain. Oh no, sorry, other way. Fab region. FC region, okay. Hinge regions. Okay. That's the easy part, so I can't use that. 5 is the FV region. I don't even know what that means. All right. Oh man, it's the afferent, bro. What? Oh. Bro. Do you guys have a way to remember this? The afferent and the efferent lymphatic vessels?
No clue.
I
I
just
think
guessed because I had the same question, but I had it with A is afferent is approaching, so A. I saw that arrow going in. I was like, okay, cool, that's afferent. Efferent is exiting.
but isn't afferent away and then efferent
No.
towards?
Afferent is approaching. Arteries are away.
I think I know what you're talking about, Shab. But I think that is it like the same, but it's different for lymphatic vessels, so maybe. Because I think you're talking about the. I think you're thinking.
Yeah, no, you're right. Afferent is towards, efferent is away.
Oh, is it? Shit. Okay.
Yeah, that makes sense. Like A is going in, E is going out.
Yeah.
Okay, so that's what the lymphatic.
Peyer's patches were small intestine.
Is it in all the small intestine, or is it like specific? Like, because I think it's like specific.
What, like
Jejunum, duodenum, something like that. Is pie patches the immune one? No, it's not, right? Oh. Okay, this one, bros. Like,
stop sharing.
Yeah.
Oh, that's an L.
How?
You got all the hard ones, man. I got like right atrium half the time.
Bro, that would have been awesome, bro. But I thought this was medial because it's like in the middle. Because I know it's one of these three, right? Is it? This is a papillary. This is 1, 2, right?
So this is
What's
no,
the
that's that. Yeah, you should have got. I think you should have got that.
I thought it was this one.
But yeah.
Or is this
no,
not even a papillary muscle?
I actually don't know. It's a hard one.
What's
Do you know
this
what
V
that?
stand
What else
for?
that could be? The must be ventral. I don't know.
Ventral. Okay.
What could the V stand for?
No clue. Either ventricle or ventral.
Hmm.
Ventral means front, right? So
Yeah.
okay. Yeah, this one.
Carl, you could have hit that 60%.
I could have, but bro, I don't know. This is the left atrium. I mean, left ventricle, right?
I think that's right ventricle.
Oh, that's the right. Oh shit.
It might be right.
So if that's the right ventricle, this is the right atrium. Then this must be the left atrium.
No, I think you might be right. I think that might be the left ventricle because it looks too big to be the right ventricle.
Okay. Oh, do we have to use this thing maybe? If that's the ventral, so it's the front.
What? No, the thing should say what view you have. What did it say? Go up. Well, it doesn't say anything.
Nah.
That's crazy.
But this is the aorta?
Yeah, I think that's the aortic valve or the yeah.
Aortic valve. So then left atrium. Actually, yeah, kind of makes sense. Yeah, okay. Yeah, okay. Nah, I need to go through some radiology again. But
Hmm.
the Clostridium one. Yeah, this one, bro. It's lucidum. Pin B. I thought it was. Why did I say this? What the?
Or B.
I was. I don't know.
I get why you might have said it, but yeah.
I was like, nah. I got No, that's a bad guess from my part. I should have stuck with this, man. To be honest. Yeah, this one's the other one. This is the other one I could have got right. This was an easy one I should have got right. I should have just looked at, because I knew it was ciliated, but I don't know why I didn't look at this closer. What? I got this wrong. What is it? Mitral valve? Shit.
My guy.
That's an easy mark I could have got right.
Wait, what is that? Let me see.
Yeah.
What was the question? E is mitral.
Oh,
No, wait. Oh, because it's left. Yeah, you looked at it as right.
Yeah.
Crazy.
I could have. I could have got this easily.
What lacking, bro?
Yeah. It's all right. Whoa. Yeah. Okay.
And then you went on a run.
Yeah. Oh, it was B1. Okay. So that, because I knew it was water soluble.
Oh, that's cool.
And then E and K, they're fat soluble, right?
E and K?
Yeah, vitamin E and K.
A, D, E, K?
A, D, E, K. Yeah, those are the fat soluble, right?
Hmm.
Oh, I guessed this. I got this right. Damn, okay.
Reverse action of phosphofructose kinase. Okay.
That's insane. So we had to know the enzymes as well. And the bruh. All right. Now I'm
Not
pretty
bad.
happy. I'm pretty happy.
Yeah,
I
that's pretty good. Pretty good. Pretty good.
Pretty happy with that. Are we doing short answer?
Nah, yo, I'm too dead to do that.
No. All right. Solid 5 hours. What were we saying? What were we saying? Anyway, we'll start. Shahab, I'll start making those quiz shorts, so we can pump those out.
Done.
I was thinking there'll be like each one will have like 5 levels. It's like it starts at like 5th grade, then it's like 10th, then it's like pre-med, then med, then like 2nd or 3rd year, and then doctor level, something like that.
Done.
Or maybe it could just be one question per video. We'll see.
Holy. I think I'm getting mucus problems, bro. I gotta check for cystic fibrosis.
Right.
Nah, no, surely not.
Lick
I think
your
it's the
skin.
vaping.
Hmm?
No, I'm good.
Salty? You're good.
Vaping?
Not salty whatsoever.
No.
All right. Let's cut it off before it hits the 5-hour mark.
Yeah.
Done.
Okay, Ka, thank you for joining us. And thank you, Shahab, for the words of wisdom, as always.
Yeah, bro. Don't say that, man. I fumbled so hard on that quiz.
Maybe you just need to get some sleep.
I'm hoping that's the case.
Okay. All right. Thank you guys for joining us. Thank you to our #1 Twitch fan. And thanks to
Much
Addy.
love to this guy.
Yeah. Our guy Addy. All right. Bye-bye. I'm out.
All right, see you, bro.